Hello, everyone.
Hi, good morning, everyone. Good morning, Dr. Suji. Okay, good morning and welcome to everyone for today's webinar on epigenetics, decoding mysterious journey of stem cell in making our body. And today we are organizing this webinar by Faculty of Applied Sciences, AIMS University, in collaboration with the Malaysian Society of Biotechnology, Molecular Biology and Biotechnology. And I'm the moderator today. I'm Dr. Venkatasuresh. And today's speaker is Dr. Suji Akarwal Singh. And very happy to introduce my friend, Suji. She received the PhD from University of Münster, Germany, working on mechanisms of leukemic transformations and epigenetic regulations and function of oncogenic networks. After that, she moved to University of Copenhagen for her postdoctoral studies and joined a renowned Institute for Epigenetic Research, BRIC, Biotech Research and Innovation Center, where she gained the knowledge, expertise in epigenetic research, understanding the epigenetic regulation in stem cells and differentiation. Now, she further moved to the University of Cambridge and joined as a Department of Hematology at a Stem Cell Institute, where she is combining her knowledge of expertise on epigenetics and leukemia research, continuing to understand the epigenetic mechanisms that are corrupted in hematological malignancies. And she published several articles in high impact factor journals throughout her career. And she's the founder and organizer of Cambridge Cancer Epigenetic Club. And Suji is a member and organizer of a Young European Hematology Research Meeting, Young EHA 2022. Suji is also serving as a guest editor for journal Cancers, which is very high impact factor. And she's now working on various projects include understanding the molecular functions of mutations in epigenetic modulators and transcriptional factors on chromatin landscape and their involvement in leukemia disease. By using various advanced techniques like gene editing by CRISPR-Cas, genomic approaches by ChIP-seq, transcriptome profile by RNA-seq, and the 3D genome organizing by DNA interaction method as 4C seq, high-seq, MEATAC-seq, and so on in vitro and in vivo models. And very, very advanced and useful area for every one of us to know more. And without further ado, I would like to happy to introduce the speaker of today, Dr. Suji Akarwal Singh.
Thank you, Suresh. Thank you for a lovely introduction. Hello, everyone, and good morning to you all. Although it's midnight here in UK, but yeah, this is the advantage of virtual platforms, that we can connect to each other despite of having different time zones and having no travel commitments. And I think this is the only positive outcome of the pandemic, that we begin to realize the importance of these webinars and virtual seminar series. So thank you very much for having me and giving me this opportunity to share my work. And I will just share my screen, just a second. I hope it's okay now.
Yeah.
Okay. So thank you very much for having me and inviting me today. And I'm really excited and pleased to share, pleased to talk to you about epigenetics and share that why I find this field so fascinating. I will also discuss some of the work I have done in the recent years in the stem cells and understanding what epigenetic regulation means in stem cell differentiation. So epigenetics, what is epigenetics? I think you hear this word very often and you read it now everywhere in magazines, in research magazines, research articles, or academic articles, or when you talk about health, diet, nutrition, diseases, you hear word epigenetics. And what is epigenetics? In the next few minutes, I will convince you that this is one of the fastest growing field of biotechnology and biomedicine. And every day we are learning more and more about epigenetics, that how it impacts our genes and how it impacts our genes that are related to the phenotype, like that is translated into the diseases and health and environment. And we are also learning that how can we use epigenetics to treat different diseases and revert the phenotype which we see. So we'll talk all about it during this session. And during my talk, I will talk to you about different definitions or different ways what is epigenetics and different examples of epigenetics, what we see in the daily life and various cellular contexts, and how our lifestyle and environment is impacting on our genes, the way genes are expressed, and how it is affected by different environment in our lifestyle. And we'll also talk about epigenetics at a molecular level, that how and what it means in terms of molecular biology, and why and how epigenetic mechanisms are important in diseases like cancer. And I will also very briefly talk about current techniques that are used to study epigenetics. And then I will discuss some of the work I have done in the recent years in stem cells. So epigenetics can be, the first definition of epigenetics came around 80 years ago. So in 1942, Conrad Weddington came up with the definition of epigenetics. And since then, many other groups and many great pioneers in the field have defined it time to time and updated the definition, which is basically the study of the changes that affect the gene function. So for example, this is my favorite one, where the study of epigenetics is the study of heritable changes that passes from one generation to another and in the gene function, but that is not without changing the DNA sequence. So the changes that are impacting on your gene function, but there is no change on the DNA sequence. And what it means, I will discuss in the next few slides. So what is epigenetics? And as I said, that Conrad Waddington in 1942 defined or coined this term epigenetics, and he gave this first definition. So epi is a Greek word, which means over, above, or on top of. So that means on top of genetics is the epigenetics. And we all know about genetics that talks about DNA chromosome, our genes, and genetic sequences. But epigenetics is on top of genetics. And what that means, I take it a very, very basic example. And this is how I define epigenetics, that if we say genetics is a base of a cake, then epigenetics is the topping of the cake. So that means if you see these three different examples of the cake, excuse me, they all look different and they all have a different appearance, different form, and different taste also, different flavors. But that is coming from the different toppings that is given to the same cake base. So imagine that this is the same base, which is behind all these three cakes, but they all have a different appearance because of different coatings, different toppings. So genetics is the same. So that means that if genetics is the base of the cake, the epigenetics is the topping of the cake. And this is how I define epigenetics, that genetics is the genome, our genome is the same, but epigenetics is the way that define or decide whether our genes are expressed or not. So without changing the genomic sequences, the epigenetics can still dictate whether the gene should be expressed or not. And that is giving the phenotype or the appearance of the individual or the way we are. So saying that study of heritable changes that are not caused by changes in the DNA sequence. So there are two main components of epigenetics. One is DNA methylation and histone modification. I'll come back to that in the next few slides, but before that, I just want to introduce one more term, which is this very classical picture of epigenetic landscape. And this was defined again by Conrad Waddington in 1957, where he proposed that epigenetic landscape is a process of cellular decision making. So by this rolling ball model. So if this is a cell which has to decide whether to go for this trajectory or that particular trajectory, that will decide its fate. So whether this cell wants to become a muscle cell or a bone cell or a blood cell, it has to take one trajectory to make one particular path and one particular type of cell. And that is decided by epigenetic landscape. So the epigenetic landscape of this cell will change if a cell decides to go to this particular trajectory. That will make it a bone cell. But if it wants to decide to make a muscle cell, it will take a different path. And that means its epigenetic landscape will be slightly different, and then it will follow this path of the trajectory. And in the past, we always thought that this process is irreversible. So if a cell has become one particular kind of cell, it stays there. But now, with the current knowledge, we know that we can do transdifferentiation. So one particular cell, which is defined to do a particular function, can still be transdifferentiated into another lineage by expressing some different transcription factors or growing it through a different media. And even if a cell can be reprogrammed back to the stem cell, so after cell has taken a particular decision and become a differentiated cell, it can be reprogrammed back to the stem cell. So this is the epistem cell technology or induced pluripotency, where you can again make a stem cell from a differentiated cell. So this is all advancement of the epigenetic modulations that you can play around. But what I mean to give you a message in this slide is that when a stem cell or a particular cell is taking a special path to become a specific cell type, and that is defined by the epigenetic landscape that will be changed. And what it means. So let's talk about some examples of epigenetics we see all around us. So if you see in this picture, you're seeing many different people, and they all look different because they all come from different continent, different climate, different race, and we all have a different appearance. But if you notice that we all have a same genetic composition, we all have 46 chromosomes, and meaning that we all have same DNA and genetic content, but still we all look so different because our epigenetic is different. And this is a very, very easy example which you see in your daily life, that how people individually look so different in appearance, although our genetic content is same. It's mainly because the genes are expressed differentially, and that gives a different appearance to us. Another example, at the cellular level, you can see that the whole body is made of different cell types, different tissues, but they all originated from one single stem cell. And this single stem cell had a same genetic component that is then differentiated, but given to the different cell types. So if you look in within each of these cell types, it always have the same genetic content and same number of genes that are present in all of these cells, but they all have a different function and different appearance, because that some genes that are expressed in this particular cell are not expressed here. So the cells which are expressed in your insulin producing cells are not expressed in your brain cells, or vice versa. The cells that are responsible for your blood formation express different kind of genes. For example, hemoglobin, that is not expressed in your neurons. And this differential gene expression pattern decides the cell type specificity or cell type identity, although other genes are also present in all the cell types. So this is just to give you an example that single stem cell that give rise to many different cell types within the body have the same genetic content, but they all have different function because epigenetics decides that. Another example you see in the daily life is identical twins. So the identical twins have, again, the same genetic content. They derive from same mother, same womb, same embryo, and they have exactly same genetic content. But later in the lifetime, you can see that they grow differently. And then because of different environment, different nutrition, different living habits make the appearance so different between even the identical twins. And this is, again, the environment that is impacting on our genes. So to summarize that, I would say that life experiences alter DNA, not necessarily its sequence, but rather its form and structure, including the chemicals. So the chemical modifications that are decorating your genes, and also it defines how the genes are, or the DNA is packed around the cell, inside the cell. And I will talk to you in more detail what it means exactly by this definition. Before that, I want to all the examples which I gave you so far were more general examples which you see in your daily life. But this is a scientific example, which is a very classical example of epigenetics, and it's called agouti mice example. And what is here shown that these both mice, as you can see very obviously in the picture, that these two mice look very different. Although you have to trust me that they both belong to the same strain, they have same genetic content, But what is the difference between these two mice is an expression of one single gene that is called agouti gene. So the mother of this mice was fed with a diet that contains folic acid, and folic acid provides the methylation donor. That means that the methylation presence, when the diet contains methyl donor, it leads to the methylation of one particular gene called agouti gene, and methylation is turning this gene off. And that is why when this gene expression is off, the mice color is normal brown, and mice is healthy and normal. But if the mother was not fed with the folic acid diet, that means there was no methylation happening on this particular gene, which is called agouti gene. This gene was expressed highly in this mice, leading to the change in the fur color. And also this gene regulates the insulin, and therefore it leads to the obesity, diabetes, and many other diseases like cancer in this mouse was prone to develop cancer. So you can see that just by expression of one gene, the phenotype of these two mice are changed. And that expression is just depending on the methylation status of this particular gene that is coming from the diet. So you can see that how the diet can influence and impact the expression of particular gene that results in the phenotype that can relate to the diseases or even the appearance of the genome of the individual. So not only the appearance, we can also talk about psychological behavior that is also depending on the epigenetic changes. This is a very nice example, as you can see here. It's the example on the rat. So if the rat was grooming and nurturing its pups or not, so that is defined in this picture, in this scientific example. So this rat was not grooming or nurturing its pups. And because of that, there was a high anxiety level in these rats. And when they discovered and they looked inside the gene expression pattern, we found that these mice, which were not cared at their beginning of their life, had lower expression of glucocorticoid receptor. And lower expression of glucocorticoid receptor results in higher level of corticosteroid hormone. And that is a hormone for high anxiety. Whereas this group of rats, which were properly cared, taken care, nurtured, and groomed by their mothers, they have a higher expression of glucocorticoid receptor, resulting in the low anxiety hormone, low corticosteroid level, results in the low anxiety in these mice. And this is how people say that even the psychological behavior was defined based on the expression of certain gene, which is again influenced by epigenetic changes. So as we can see that if I can summarize all those examples which I talked to you just now were like simple things like environment, diet, nutrition, stress, or physical activity have impact on our genes without influencing the genetic information. And external factors can impact your genes via epigenetics, and that results in the phenotype, what we see. So now let's go back to I'll take you to the molecular side of epigenetics. And first of all, just to go back to the basic books, and we talk about transcription and translation before we try to understand epigenetics at molecular level. So we all know that this is a DNA, and DNA contains different genes and different genes that when genes are expressed, that means DNA is transcribed into the RNA by a process called transcription. That means this gene is expressed into RNA. And this RNA then will be translated into a protein, a functional protein. So transcription and translation leads to the production of a particular or functional protein from the DNA or from the gene. Now, whether this gene is allowed to make a protein or whether if this gene is not allowed to make a protein, this is decided by epigenetics. And what I want to say here, that addition or removal of a small chemical tag to the DNA or to the gene will make whether gene should be on or not expressed. So imagine that when I say chemical tag, that means if there is a sticker, if we imagine this tag as a post sticker that is pasted on top of this gene, that will dictate whether this gene should be on or off. For example, if there is a label on this gene as an on signature, that means this gene will be transcribed into RNA and make a functional protein. But if there is a tag of off signature, that means this gene should not be transcribed and not translated and not make a functional protein. And these tags are epigenetic tags. And what are these tags? This is what I'm going to tell you now. So we know that, and before we go there, we have to understand DNA and chromatin. So we know that DNA double helix model, we know that DNA is a two-strand, but this DNA is wrapped around histone proteins, and histones are the octamer of 4 different proteins. So H2A, H2B, H3, and H4. And that makes a octamer of histone protein. And around that histone, the DNA is wrapped around. So this is how it's called chromatin. So when DNA is wrapped around histone, it's called chromatin. And many chromatin structures come together. So many histones and DNA fragments come together. And this is called a bead on string model. So this is the chromatin. So this is how DNA is compacted inside the cell, because it's wrapped around histone proteins, and then it gets more and more compacted to make a structure which is very tight and can fit within the nucleus of the cell. So if you look within the histone protein, which is basically octamer of 4 different proteins, have histone tails that are protruding out of this ball. And these histone tails are the regions which are subjected for chemical modification. That means these histone tails get a chemical tag of on or off switch, which I was talking about. So epigenetics have 2 main components. One is DNA methylation. So the DNA itself can get methylation tag, and that dictates whether gene should be repressed or off situation. Or the histone tails also get a tag of switch on or off gene, which is the small tails which are coming out of the histones. So if I show you in much more detail about the DNA methylation, what it means, that if you remember that DNA methylation, you remember the structure of DNA, you have 4 bases, A, T, and G, and C. So C is referring to the cytosine. And DNA methylation means that there is a methyl group added to the 5th position of the carbon in the cytosine ring. So this is the, if you look at the structure of cytosine, then methylation is added to the 5th carbon of this ring. And this is called methylated DNA. And this whole enzymatic reaction is catalyzed by an enzyme called Nnmd, the DNA methyltransferase, that transfers the methyl group from a SAM cofactor to the 5th position of the carbon. And this is how the DNA methylation is taking place onto the on the DNA. And wherever you find the C or the cytosine methylated, that means it gives a signal for an off switch. So the genes that have a methylation on its DNA, that means those genes should be shut down and should not be expressed. And this tag is reversible. So there is another DNA methylation inhibitors that can remove this tag, and then this gene can be transcribed and switched on. Now, if we talk about another histone, another epigenetic modification, which is histone modification. And if you remember this slide here, the picture where I said that there are histone tails that are coming out of the histone ball. And those are the regions where the epigenetic modification is taking place. And this part is zoomed in here. So this is one of the histone H3, and this is the tail that is protruding out. The tail is basically 79 to 80 amino acid sequence, which is coming out of this protein structure. And within this tail, there are different amino acid residues that are modified by chemical modification. So they are gaining methylation or acetylation or phosphorylation. So these are covalent modifications which are added to these specific amino acid residues onto the histone tails. Now, the important part here is that the position of the amino acid that is modified, that is the important message here. So if there is a lysine, K represents lysine amino acid. So if the fourth position, the fourth amino acid of lysine is methylated, that brings a tag of on, switch on condition. That means the genes that have a methylation at fourth lysine should be turned on. Whereas if there is a methylation on 27th position of lysine, that means it's a tag of turned off situation. And similarly, lysine 9 should be methylated. That means the genes should not be expressed and it should be off. So this is the on and off situation decided by the methylation status of the lysine residue on a histone tail. So altogether, this combination of different histone modifications is called histone code. And the combination together decides whether genes should be expressed, genes should be on and transcribed, or whether it should be repressed or silenced. Now, as you can imagine that this is a methylation, acetylation, phosphorylation, that means it's a chemical addition, and that has to be done by an enzymes. So there are different enzymes that can do these modifications. And in epigenetic field, we call them writers because they can write the modification on these particular residues. So there are histone acetyl transferases that can transfer the acetyl group to particular residue. There are kinases that can put the phosphorylation group on a specific serine or threonine residues. There are also KMTs, which are lysine methyltransferases that can do the methylation of the particular lysine residues. So there are many different writers. Then there are also readers. That means the group of proteins that can read these modifications and then translate that information into the complex or signal that whether this gene should be transcribed or not transcribed. For example, you know that the transcription requires the RNA polymerase to bind. So RNA polymerase can only bind if there is a lysine 4 methylation present on the cell, on these histone residues. But if there is no lysine 4 methylation, instead there is K9 methylation, which is read by many other readers. That means a full complex of proteins present here. They will block the binding of RNA polymerase, and therefore the transcription cannot proceed. And therefore the genes will be silenced. Now, there is another group of chromatin modifiers that are called erasers. That means they can remove these modifications. And this is the best part of epigenetic system, that it is reversible. So histone deacetylases are the group of proteins or enzymes that can remove the acetylation mark from the histone tails. Similarly, there are demethylases that can remove the methylation group and revert the situation. So the genes that were repressed or shut down or off in one situation can be activated. So it's not a permanent situation. So epigenetics is a reversible situation, and that is why it's one of the very attractive target for the therapies, because you can revert the situation if something has been inappropriately silenced or activated. So now here I want to show you some example, one more to understand that euchromatin is a status on the chromatin, which is means that transcription is on. That means active transcription is taking place at the euchromatin region of the chromosome. That means if you look at the molecular level, that means this region has a lot of acetylation, and acetylation is done by histone acetyltransferases. So that means writers have done the acetylation, for example, P55. Now, this acetylation is read by a group of proteins that have a bromodomain, which is called readers. So set of proteins can read this modification and build a protein complex around it. That means it will dictate the information to RNA polymerase so that it can lead to the transcription of particular gene. Now, this acetylation can be removed by H-tags, histone deacetylases, and revert the situation. Similarly, if you look in the repressed state, that means heterochromatin area of the chromosome, where gene transcription is usually off. That means there are a lot of methylation at the lysine 9 residue, and that is done by enzyme lysine methyltransferase. One example of that one is SUV39H1, and this enzyme can lead to the K9 lysine 9 methylation. Now, this methylation is read by a group of proteins which have a chromodomain, and that's how it builds a complex of protein around this area and leads to the silencing of gene expression. One more example here is the enzyme complex that can do lysine 27 methylation. And this is a group of proteins called polycomb repressive complex. Within the polycomb repressive complex, EZ-2 is the enzymatic subunit that can methylate the K27 residue on the histone tail. And when this methylation is happening, the whole complex of polycomb can sit here on this area. It can also recruit histone deacetylases that can remove the acetylation, and this whole region will become completely silenced. That means the gene expression will be shut down. The polycomb repressive complex can also bring DNA methyltransferase into the vicinity, and that's how it can also methylate the C cytosine residue of the DNA that leads to the complete silencing of the gene expression. So this is how you can see that putting a chemical modification on a specific residue can result in the situation that gene should be turned off or on. Now, this gets more and more complicated. So the examples I have been giving is in a very simplified form, but it gets complicated because each residue can be methylated by many different enzymes. So depending on the cell type or the situation or the locus, there are many enzymes that can do the same modification. So the green ones here are the writers, that mean methyltransferases. And you can see that MLL is a methyltransferases that are responsible for lysine 4 methylation. And there is a set of them that can do this job. But then they are always dependent on cell type. So some cells express MLL1, whereas other cell type will express MLL3. In the same way, the histone, the purple ones are the erasers that can remove the methylation status of lysine 4 methylation. Now, orange ones here are the readers. There are many different kind of proteins that can read this modification and then transfer the information to the next complex and leads to the gene activation. So this is a complicated situation in a way that there are many different enzymes that can do the same job, but then there is a redundancy in the cell type, or there is a uniqueness depending on the situation. Now, this whole thing can be, if this whole thing is, as you can imagine, that this is getting very important process for the development. And if there is any misregulation, that can lead to the abnormal expression of the genes. And this will result in the aberrant gene expression, which is the situation in cancer or many other diseases. So for example, if there is a higher expression of one of this protein, for example, MLL, let's say if MLL is highly upregulated, it will inappropriately methylate the lysine 4 residue in a lot of other genes which should not be methylated. And therefore, it will result in the abnormal expression of the genes. Or if there is higher expression of this one of this enzyme, which can do the repression or the silencing of the gene expression, this will result in the inappropriate or abnormal silencing of the lot of genes. And this will result again in the situation which is not normal. So either cells will proliferate because there is no control and the controlling genes are off, or cells will divide inappropriately. So there is also a situation, so in cancer, this is all the situation that's happening in cancer. For example, there is a mutation that means the lysine 27 residue of histone tail is not present in some of the cancer cells. And that is the situation in glioblastoma, that in the glioblastoma, lysine 27 is mutated, and therefore there is no methylation happening on the lysine 27 residue, resulting in the abnormal expression of the genes, which results in the abnormal proliferation of the cancer cell in glioblastoma. Similarly, there are other enzymes, the erasers or writers or readers, they are abnormally expressed in the cancer situation, and that will result into the abnormal expression pattern of target genes, and therefore the disease status will come. For example, if I talk about acute myeloid leukemia, the leukemia status, the blood cancer, the expression or the mutation is observed in many of the writers. So these are few examples of the writers. These are the enzymes that do the epigenetic modification. For example, P300 or CBP is a acetyltransferases. And in acute myeloid leukemia, there is a mutation observed in these genes. That means these genes are inactivated because of the mutation. So when there is a mutation in acetylation writers, that means there is no acetylation happening on the genes where it should be acetylated. That means the target genes are shut down forever. And therefore, it results in the abnormal expression of genes and the disease status. Similarly, in leukemia, there are erasers like hashtag histone deacetylases are abnormally expressed. So they are highly upregulated. When histone deacetylases level goes high in the cancer cell, it leads to the abnormal deacetylation of the whole region, which will result again in the abnormal expression of the genes and will result in the cancer situation or the unlimited proliferation of the cells. So this is how different mutation in the epigenetic writer, reader, or erasers leads to the abnormal expression of the genes and in the situation of the uncontrolled growth, uncontrolled proliferation of cancer cells. But the important part here is that we can control this situation because epigenetics is reversible. So for example, there are DNMT inhibitors and inhibitors that are in clinical trials that can stop the activity of DNA methyltransferases. That means abnormal methylation in a cancer cell can be controlled by using a drug called DNMT inhibitors. And these drugs can lead, so for what happens in this situation, that if there is a abnormal methylation of certain genes, which are tumor suppressor genes, that means the genes that should control the growth of a tumor are abnormally silenced. They are shut down, they are not expressed because there is a higher methylation of these genes. Now, if we put a DNA methylation inhibitor onto the cells, that will revert the methylation situation. The tumor suppressor genes will be re-expressed and will stop the proliferation of a tumor cell. And this is how epigenetic inhibitors or epigenetic drugs are useful and are already in a clinical trials for many different malignancies. Similarly, HTAC inhibitors are also in clinical trials that can revert the acetylation situation in particular cancer or other diseases. There are also methyltransferase inhibitors, and that can revert the methylation situation of a cancer cell where oncogenes are highly expressed because they have higher methylation at lysine 4 residue. And this situation can be reverted by inhibiting the activity of these histone methyltransferases. And there are also BET inhibitors that are bromodomain inhibitors that are inhibiting the protein activity which recognize the acetylated regions. So this is just an example to show you that a lot of epigenetic drugs are already in clinical trials because they can revert the situation of abnormal epigenetic regulation that has been identified in many cancer cells. Now, I just want to talk very briefly about the methods that we use to measure epigenetic modification. So as you got an impression that there are many different kinds of epigenetic modification, there are DNA methylation, there is a histone modification, and to understand that, we have to read those modifications. And in order to read DNA methylation or epigenetic modification, there are different kinds of sequencing patterns, sequencing methods. One is called chip sequencing, like chromatin immunoprecipitation, followed by next generation sequencing. And then there are methods that can detect the chromatin compaction, so that how close these nucleosomes are present. And then there are the methods that can decide if the genes are too compacted because they are too close, or if the genes are more in the open area because nucleosome occupancy is not that dense. Then there are methods that can detect the looping pattern of the chromatin because as you can see, the DNA is found around and it's wrapped around the nucleosome, but again, it is compacted by a looping structure. And the looping of DNA is also very important for bringing enhancers and promoter close together. And there are methods that can detect the looping of this DNA of the chromatin by chromatin conformation capture assays like 3C, 4C, high C, those kind of methods. So there are a range of methods that you can do to detect epigenetic modification. And then on top of that, you can look for the RNA expression. That means that will define whether your genes are transcribed or not. So you can measure the amount of RNA expressed in particular cell. So by overlaying and by integrating all these analyses on top of each other, you can get a reliable and useful information, and you can actually generate an epigenetic landscape of a particular cell type. And then you can decide and say that whether this particular cell has this kind of epigenetic landscape, it has a methylation on these regions, it has a histone modification on these areas, and therefore these genes are expressed and these genes are not expressed in this particular cell. So this kind of multidimensional epigenetic profiling is very important to understand the landscape of a cell type. I just want to give a very brief example of one of the technique, which is called chromatin immunoprecipitation, followed by next generation sequencing. It's called ChIP-seq. So what we do in that method, that we cross-link the cells. So we cross-link the cells means we put the cross-linker like formaldehyde onto the cell that will fix your DNA and the proteins that are in the vicinity. And after cross-linking, you shear the protein, the chromatin, you fragment the DNA. So that means your long DNA is fragmented into small fragments, and the proteins that are close or bound to it are already present there. Now you put an antibody against your specific protein of interest, or it could be a modification. For example, if we are looking for histone methylation, we put an antibody against this histone methylation, and then we pull down this specific area. a specific protein complex, which contains the DNA of your interest. Then after purifying the DNA from this complex, you can sequence the whole through next generation sequencing and align the profile onto the human or mouse database. And you can see that you can see the pattern, and you can guess that you can identify the specific histone modification because you have used the antibody against that specific modification. In the same way, you can also look for the binding of a transcription factor. For example, if a specific transcription factor is bound to the DNA regions of a particular gene. So you can do a chip sequencing using an antibody against your transcription factor to see whether it's bound to what regions of the genome. Now, there are a lot of limitations in the chip sequencing technology, which had been here in the field for about 15 years now, that you require a lot of cells, a lot of material for performing this technology, and it takes longer time, and the quality of the reads are very low. But now, within last 2 years, I think there are much advanced techniques like cut and run or cut and tag that has much lower requirement of the cell material, and it's much faster to perform the experiment, and also the quality of the data is much better and higher. So that's just to give you an impression that even the genomic techniques are getting advanced and improved with the time. And it's getting easier and feasible to use the primary patient materials or the primary materials for performing such kind of analysis and experiments. So before going into the data, I just want to share some more points that epigenetics field is very fast growing, and there are different branches of epigenetics that one can study. So we already talked about how genes are turning on and off, and how cells are having a specific identity. How do they maintain their identity based on epigenetics? But one important point of epigenetics is also DNA packaging. That you can say that we all know that the DNA within one single cell, the length of a DNA is 2 meters, and the 2 meter of DNA has to fit into the 2 micron nucleus. The size of the nucleus is 2 micron. So you can imagine that how higher degree of compaction is required to fold this 2 meters of DNA to fit within the 2 micron. And this is happening with this chromatin compaction, what I have been showing you, that DNA is wrapped around histones, and then it makes a very compact structure. And this is one of the field of epigenetics, which people try to understand that the compaction and what controls the compaction and other factors. And again, the diseases that impact of or the effect of epigenetics on different diseases and how and where it impacts. So now what I wanted to show you in the last few slides, that some of the work which I have done in the recent year about how genes turn on and off in a stem cell, and how the differentiation is, the epigenetic landscape is changing during differentiation. And I want to show you by showing you the example. So let's skip that part, because I think I've already talked about this slide. But as we know that a single stem cell can differentiate into many different cell types within the body. And this differentiation process is regulated by epigenetic landscape. And this is what I want to talk to you, that what it means. So imagine if this is a stem cell and it has multiple genes, but all genes are not expressed within the stem cell. Some genes which are not required will be shut off by epigenetic modification, which we know now that it's could either be methylation of different residues. Now, some genes will be turned on in the stem cell, like genes that are required to proliferate or maintain the stem cell, which will be expressed here, and other genes will be turned off. Now, if that stem cell decides to make a nerve cell or a blood cell, the gene expression pattern will change. So the gene which was active in the stem cell will be shut off in the muscle cell, but other gene which is required for muscle, like muscle contraction and all that kind of function is required for a muscle cell, those genes will be turned on, whereas other genes will be turned off. But if the stem cell is deciding to make a blood cell, for example, red blood cell, the expression of hemoglobin gene should be turned on, which was not required in the muscle cell. So that gene will be turned on, whereas other genes will be turned off. So this is how differential gene expression will decide the cell identity, whether a stem cell will decide into which lineage, and the different expression pattern will be changed onto the in different cell types. And that will be decided by different histone modification or epigenetic modification. One more thing I want to talk to you about is enhancers. The enhancers are the cis or the gene regulatory elements on the DNA. And these regions have a potential to dictate a message to a particular gene, whether this gene should be on or off. And these are like, you definitely have known about it in the molecular biology lessons, that enhancers can be present far, far away from a particular gene, but then they can go and they can come into the close contact of a gene promoter and then dictate a expression message for a gene. And these enhancers can be recognized by different epigenetic modifications. So this is now also well defined, that active enhancers contain histone 27 methyl acetylation and methylation mark within a region. And by detecting those modifications, you can identify enhancer within a cell. And enhancers have a tendency to come close to the gene promoter and regulate the expression of a gene. So what we did in this study, that we took a mesenchymal stem cells from human bone marrow, and mesenchymal stem cells from adult human bone marrow have a potential to differentiate into bone, cartilage, muscles, or the adipose or the fat tissues. So mesenchymal stem cells are called multipotent stem cells, and they can give rise to multiple lineages of the different cell types like bone, cartilage, muscle, and adipose tissue. And mesenchymal stem cells are the very important source of regenerative medicine, because they can be isolated from adult bone marrow and they can be modified artificially and then injected back into the patient. And this is the research that's going on to improve the many disease conditions nowadays. So to understand this, to have a better understanding of a mesenchymal stem cell differentiation at the epigenetic level will be very helpful for a regenerative medicine purposes. So what we did in this study, that we took a mesenchymal stem cells and profiled the epigenetic landscape during the bone formation into the osteogenic lineage. And this is shown here, that on the left side here, you can see this is the mesenchymal stem cells. And when you differentiate into the bone lineage, it takes about 21 to 28 days to make a complete bone cells or mature bone cells. So we profiled the epigenetic landscape at early stages of differentiation at day 2, 5, or 10, and wanted to see that how the epigenetics is changing when cells are differentiating. And to understand that, we have done a profiling of histone or lysine 27 methylation in these cells while they are differentiating, and also looked for the expression of RNA to see what genes are expressed, and also looked for K4 or lysine 4 methylation, which is an active methylation mark. So by doing a lysine 27 methylation, if you remember from previous slide, this is a marker for a repressed or a silent status. So we identified that one particular locus, for example, ZBTB16 or PLZF locus was demethylated. So we found that the K27 methylation was removed or lost when cells are differentiating towards the this lineage. So if we zoom it into within this locus, which is shown here in a profile, I will guide you through this picture here. So this is down here is RNA sequencing data, which is the top is a stem cell, so the mesenchymal stem cell, which is a day 0 status here, and it is showing the abundance of that RNA. The RNA, that means that is transcribed for this gene, the PLZF gene, which is expressed, which is not expressed in the stem cell. As you can see, you do not detect the RNA for a PLZF gene. But when cells are differentiating for 2 days or 10 days, you detect the expression of this gene. So you detect the RNA for this gene, which is expressed. So that means this gene was not present in the stem cell, but it started to express. Now, when we look at the epigenetic state of this gene, here, if you focus on this part of the column here, it says lysine 4 trimethylation. And if you remember that lysine 4 methylation was an active or switch on situation. So in the stem cell, there was no lysine 4 methylation at the promoter of this gene. But when cells are differentiated for 10 days, we detected the amount of lysine 4 methylation at the promoter of this gene, which correlates with the expression of this gene. That means this gene was not on, but it became on because it got the lysine 4 tag to switch on the gene expression. Now, the second panel here, it shows the lysine 27 methylation. That means the repressive mark or the shut-off situation. And you can see there was a high amount of lysine 27 methylation on the gene locus here. So that means this gene had higher amount of repressive modification, which was removed completely when cells were differentiating for 10 days. And additionally, we also looked for acetylation of the gene. And you can see that on this locus, there was no acetylation. Acetylation is again the active mark, so the gene expression mark. And you can see that in the red panel, that the acetylation was again achieved, or the acetylation was gained. So now if I summarize that part, you can see there was no K4 methylation, the lysine 4 methylation in the stem cell, but the differentiation leads to the gain of methylation at lysine 4, which is a marker for switch-on situation, and it correlated with the RNA expression or the transcription of this gene, which came at day 2. And similarly, the repressive mark was there in the stem cell, which was gone into the differentiating cell. So suggesting that how this differential situation of epigenetic modification is taking place within this gene locus, and the gene is expressed. We also detected by the Western blot to see the protein expression of this gene, and we could detect that in stem cell, this protein was not present, whereas when cells were differentiating, we saw the expression of this protein coming up. PLZF is a transcription factor, so we did a chip sequencing experiment to see where all on the genome wide, where this PLZF is present. And when we looked for the binding of PLZF, we found that it correlates with the binding of histone acetylation at lysine 27 and K4 methylation. These two modifications of histones represents the enhancers that suggested that PLZF, when it is expressed, when cells are differentiating, is going and binding to the enhancer regions within the genome. And next, we looked for the changes in acetylation pattern upon PLZF binding. And what we observed that when cells are differentiating at this stage, we found expression of PLZF that relates to the increased acetylation of those regions, and increased monomethylation, and increased expression of nearby genes. So the genes that are nearest to those enhancers were highly expressed, and majority of these genes have function on the osteogenic lineage. So the proteins that have function, either those were markers like ALPL, alkaline phosphatase, which is a marker for osteogenic differentiation. So we identified that PLZF expression was completely shut down in stem cells. It starts to express when cells are differentiating. It binds to the different enhancer regions and leads to the increased acetylation of enhancers and increased expression of nearby genes. Now we wanted to confirm whether PLZF is responsible for this increased acetylation at enhancers. And therefore, we knocked down PLZF expression in these stem cells by using siRNA technology. And when we knocked down the expression of PLZF, we saw that increased acetylation was gone. So the acetylation that was increased during normal differentiation was reduced if there is no PLZF present. And that correlated with the reduced expression of those genes that were normally upregulated. So that the osteogenic or the bone-related genes that were normally expressed upon differentiation were reduced in its expression if PLZF is not present, suggesting that PLZF is a factor that regulates the expression of osteogenic specific genes by binding at enhancers. And within that study, we also wanted to then characterize one of the PLZF bound enhancer. So we took this enhancer area, which was bound by PLZF. So this is a ChIP-seq track for a PLZF. The upper one is showing the stem cell situation, and the lower one is showing the differentiation situation. That means the PLZF is present. As you can see, this is a signal for PLZF binding at this particular enhancer. And this enhancer has a K27 acetylation mark in the differentiation situation, which was not present in the stem cell situation. So we took this enhancer element and cloned this area into an artificial reporter system. So in this reporter system, there was a GFP expression, which was preceded by the enhancer. The enhancer, which we identified from here, we took this area and we cloned it in here. And this is a lentiviral construct. So we lentivirally expressed that construct into the mesenchymal stem cells and then differentiated those cells to see whether GFP expression is on, whether this enhancer is active in the stem cells. And to understand that, we did the flow cytometry to look for the GFP expression. And as you can see here, this is a flow cytometric profile for GFP expression. GFP expression should be seen here. This is the stem cell, and as we can see, there is no GFP expression. There is no signal coming in this area in the stem cell, suggesting that this enhancer is not active in stem cells. But when we differentiated these stem cells, we saw increased expression of GFP in these cells, suggesting that this enhancer becomes active when cells are differentiating towards osteogenic lineage, suggesting that this enhancer is a latent development enhancer, which is not active in stem cell. And this enhancer only becomes active in the differentiation situation. We did different control situations and we cloned many different control enhancer regions and we found no GFP expression. Only GFP expression was observed when we cloned this particular enhancer and induced the differentiation in the mesenchymal stem cells. We also performed a ChIP experiment for many other enhancer binding proteins, which are known to bind to enhancers. For example, mediators, mediator 1 and mediator 12 complexes are known to bind to enhancers. And we found that this enhancer is gaining the binding of mediators proteins and P300 proteins after differentiation stimulus, suggesting that this enhancer becomes an active element upon differentiation, bound by PLZF, bound by enhancer proteins, and has an activity. Now, the big question for an enhancer field is that what is its target promoter? And in order to know which gene is targeted by that enhancer, we performed a 4C experiment. The experiment to see the looping of a particular enhancer region into the next or the neighboring gene. And to understand that, so by doing a 4C experiment, which is shown in this plot, and I will simplify it for you, the orange orange profile here represents the stem cell situation, whereas the blue profile here represents the differentiation situation. And what we found here that if we take an enhancer, our enhancer ENP element as a view, we found there is an interaction happening here, which is only specific for differentiation situation. As you can see, there is a blue profile coming up, whereas there was no profile in the orange situation, suggesting that this area, which is our enhancer, is interacting with this area. And if you look up here, this is a promoter of another gene, which is called Nnmd. And I can tell you that the distance between this enhancer and this gene promoter is 100 kb. So we found an enhancer that become active in the stem upon differentiation, and it's looping to the promoter of another gene called Nnmd. And then the obvious question was that whether the expression of this Nnmd gene is influenced. And we looked for the expression of Nnmd gene during osteogenic differentiation. And we found that this gene is induced in expression as cells are differentiating. Now, the question was, what is Nnmd? Nnmd is a methyltransferase. It's a nicotinamide methyltransferase, and that regulates the metabolic situation by regulating the level of SAM. So SAM is a cofactor for methylation. If you remember from the previous talks, that SAM is providing the methylation to all the methyltransferases like DNA methyltransferases or histone methyltransferases. And therefore, regulating the SAM balance in the cell is very important. So we think that Nnmd expression is very crucial during the osteogenic differentiation situation, because that's how it will regulate the methylation status within the cell. And to summarize our work, I can show you in this illustration here that mesenchymal stem cells have, we identified a locus, ZBTB16 locus, which has a repressive modification, repressive epigenetic modification by K27 trimethylation. This locus was completely repressed and off due to the lysine 27 trimethylation done by polycomb repressive complexes. Upon osteogenic stimulation, when cells are stimulated for differentiation towards osteogenic lineage, this whole locus becomes de-repressed. That means it loses the K27 trimethylation. It gains the K4 trimethylation, which is an active histone modification. It gains the K27 acetylation, which is again an active epigenetic modification, results in the expression of this gene, PLZF. And additionally, there is an enhancer present within the locus of this gene, where it is bound by PLZF, it was recruited by many other enhancer binding proteins like P300 and mediator. And this enhancer then loops to the next gene promoter, which is called Nnmd, leads to the upregulation of this Nnmd gene. And this regulates the SAM cofactor, which is a methylation provider, and regulates the methylation in the cells during osteogenic differentiation. So this was our finding overall during the differentiation of osteogenic committed cell differentiation. So I think with this, I hope I have convinced you that epigenetic is one of the fascinating field to study in the normal development, but also in the disease situation. I didn't have time to go through the work which I'm doing currently, but I would like to stop here, and I want to acknowledge my current working place, which is at Cambridge Stem Cell Institute, group of Brian Huntley. The work which I have shown you was done and while my stay at Copenhagen University at BRIC. And I want to thank you for your attention and happy to take any questions you have.
Thank you. Thank you, Suji. It was a wonderful overview of the epigenetics, starting from very basics, what is epigenetic and modifications, and that includes the writers, readers, and erasers, how that influence the epigenetics, and also included how the mutations of the writers, readers, erasers can abnormally express themselves, and that leads to the abnormal expression of the different genes, and that involved in different diseases, and the overview of the epigenetics in different areas, and then it leads to the epigenetics in stem cell differentiation, and that mesenchymal stem cells, how it can differentiate into osteoblasts. And that is a wonderful story, and retrospectively understanding the role of NNMT in indirect way using different kinds of sequencing technologies, and finding the exact what is the thing playing the role in the differentiation is a wonderful using the advanced technology. Excellent information, Suchi, and appreciate for your time. And especially, that you are at 1 a.m. now. I think now it's about 2 a.m. Thanks for accepting our request to be a speaker for this topic, wonderful topic. By this, all our students and the audience all will get benefited. And yeah, I think any participants, if you have any questions, you can unmute yourself and ask any questions to Dr. Suji Akarwal.
I would like to, can you hear everyone?
Yeah, Prof.
Dr. Suji, actually, I'm very thrilled to hear your talk. It's an unexplored area. It's really beneficial to all the students. And actually, this event is hosted by Malaysian Society for Molecular Biology and Biotechnology. The whole Malaysia is listening to a talk. All the biotechnologies are there. So it's really good. You give from the fundamental to the advanced level, and I really enjoyed listening to your talk. On behalf of the faculty and Ames University, I think sincere appreciation.
Thank you so much. It's a pleasure. An honor to be here today. Thank you.
Any questions from the participants? Either you can type in the chat box or you can unmute yourself and ask.
Maybe Dr. Suji can cover about opportunities. The student always think about going for post-graduation overseas. Many final year students are there in the audience. And actually our students, some of our students have gone to Cambridge, and there are a few of the universities in UK. They always ask for the pathway, how to go about it and so on. And you can tell about opportunities in this area, it's particularly epigenetics, but in cancer therapy and treatment, maybe you can highlight some of the points. Our students are very intelligent, but lately I'm observing, because of the COVID for the last 2 years, they have been bombarded electronically through Zoom and Team and all kind of thing. It's a kind of inhibition is going on now.
Right, that's everywhere. Yeah.
But slowly we are opening up face-to-face classes, and they are shy, but they have a lot of questions in them. I wish they will ask you today.
I understand. I think this pandemic has really affected.
Yeah.
Very negatively, I would say. But yeah.
Yeah, there is somebody asking questions. I think we can allow them, Dr. Suresh.
Yeah, Darshini, you would like to ask any question? Unmute yourself. You can ask.
Amida is asking. Yeah.
Amida.
Hello, sir. Good morning.
Yeah.
Sir, I want to ask a question. That is there any relation between chemotherapy and this epigenetic treatment? Is there any relation between this?
Very good question. Yeah, there is. So the epigenetic drugs are now combined as a dual therapy approaches. So like, if you know that chemotherapy has a lot of broad spectrum, that's very unspecific kind of therapy. So now people are trying to combine the chemotherapy together with epigenetic drugs to give a kind of boost in the therapeutic window, but also that will lower the chemotherapy level. So to give a lower dose of chemotherapy together with the epigenetic drugs, you can combination of these two will be more beneficial. So these things are in clinical trials at the moment, where people are trying to do a dual combination, combining chemotherapy together with the epigenetic drugs.
Thank you, ma'am. Thank you.
You're welcome.
Good morning, doctor.
Yeah.
Yeah, morning, doctor. I'm Darshini. I'm currently doing my PhD regarding to oral cancer. I would like to ask a question. The cell line that I'm currently using, theoretically, they mentioned that after 10 passages of my cells, the EGFR gene expressing in a very low level. So is there anything to do with the epigenetics?
That's possible that sometimes after certain passages, the gene gets methylated, and that is resulting in the lower expression of the gene. So if you know about that particular gene is lowly expressed, you can check the methylation status by several techniques like DNA methylation at the promoter of that gene, and detect the methylation if that is the reason for low expression of your particular gene.
Okay, all right. Thank you, doctor.
There is a question in the chat box from Ho Chai Ling. How specific are the inhibitors to different types of methyl transferases or acyl transferases?
So specific means, you mean if they are not redundantly affecting?
I think maybe using as a drug, I guess what I mean by Ho Chai. If you want to specifically target inhibiting different types of methyl transferases, so how specific they are?
Different inhibitors that are developed now. For example, there is a EZH2 inhibitor that's now coming into clinical trials, and that is specifically affecting the activity of EZH2 enzyme. And similarly, there are also K9 methyltransferase inhibitors that is inhibiting the K9 methylation by inhibiting SUV39H1 activity. But if you increase the dose of that inhibitor, it will also non-specifically affect the other enzymes. So I think that dose titration is something that decides the specificity at the moment. But yeah, the drug companies and the small molecule development industries are working on it to make it more specific inhibitors for specific enzymes. But there are already some out.
Okay, thank you from that. Suchi, I want to ask another question about your work. PLZF, when you knock down, right? So you have seen the different expression of things. And did you observe the differentiation from mesenchymal, or it stopped the differentiation?
It stopped the differentiation. So we did not see, so cells became very abnormal. So they like screwed up in middle of the differentiation. And we did not see the expression of alkaline phosphatase or other marker genes that should be on after differentiation. So differentiation itself was completely corrupted.
Corrupted, okay. And in a general question, maybe not related to the work you have done, the induced pluripotent stem cells, right? So the somatic cells already it is differentiated and having a lot of signatures there. So if you want to convert that, how I think it will remove all the signature from the chromatin, or how it's possible actually?
I think it's erasing the epigenetic. Totally, yeah. So it's done by re-expressing certain transcription factors. So I think if you know this Yamanaka transcription factors, that is put into the differentiated cells, and then it reprogrammed back into the pluripotent cells. And then the methylation or the epigenetic pattern is completely revert in those situations. And I think there are still certain areas of DNA methylation that is still not completely erased and recovered, like, that reprogrammed. But this is something people are working on it and trying to identify how that's been done. Yeah. But yeah, the epigenetic pattern is reversed when cells are.
And some of these Yamanaka factors are actually was observed differentially observed in a cancer also, right? It's a bit scary. I don't know.
Yeah. I think that's the disadvantage of these technologies, that people are still kind of reluctant to go behind that for therapeutic purposes. So for understanding, for a research purpose, it's still fine, but to use it as an application in the therapy, still there are a lot of
limitations.
Yeah.
So, any other questions from anyone?
Doctor, I have another question.
Yeah,
it's just out of curiosity. Cancer cells are mutated in different ways. Like different cancer cells have their different gene mutation, right? So along with our research, like when we are doing our research, is there any chances for that particular mutated gene to recover from the mutation? Means like, for my cell that I'm currently using, the P53 gene is mutated. Is there any chances for the gene to be normal back, like relapse again, be normal back? Is there any chances for that such thing to happen? Just out of curiosity.
Yeah, sure. So now by doing CRISPR, so you can do a targeted CRISPR and you can revert the phenotype. So you can mutate back. So it's called targeted CRISPR technology by putting a point mutation. So by using a CRISPR, you can either insert a point mutation onto a gene at a specific area, or you can create an isogenic cell line where you can revert the mutation. So you can make it as a normal wild type gene by using a CRISPR technology. And then you have a situation where you have a same cell type with the mutation and same cell clone without the mutation. And then you have to study both in parallel to say, by reverting the mutation, what is the impact of that gene? But it's possible now by using CRISPR.
Okay, that is when we do by ourselves, like we change the genetic modification. Is there any chance, like naturally it changed by itself while we are doing the work? Yeah, naturally by passaging and all, while we are doing, is there chances for it to gain back
I don't know, like in the in vitro situation, it may not, but in vivo, you can lose the clone. So for example, there are cell growth and then different mutations can take over. And if your mutation on particular gene is not that strong enough, it can lose the clone of that particular cell type of that mutation. This is like happening in the leukemia situation, that if it begins with one particular mutation on a gene, but after several growth or something, then certain clones can take over and one mutation is lost, but then it's taken over by other mutations. But if you are trying to suggest that the cells are automatically lost and then become normal.
Yeah, yes. Is there any chances like that? No.
I don't know.
Okay. All right. Thank you.
So any other question from anyone? Okay, so if you don't have any questions, so I would like to thank Suji for a wonderful summarizing, very simplest way she put the complex thing. It's a very complex, so many writers, readers, and erasers that nature reviews that picture itself confusing. A lot of things, but that complex things, and you made very simplest way you shown to our students, and I would like to express my thanks to Dr. Suji for spending this much time and during this midnight and compromising the timing, because we have a lot of difference timings, 8 hours difference. Either we have to conduct ourselves evening or you have to conduct your night. So you compromise that, and thanks for that. and they've covered different areas that includes the different sequencing technology. Maybe later, maybe we can again invite you, if you are available time, to brief on the different sequencing techniques like chromatin sequence, DNA modification, the immunoprecipitation sequencing, all those things. With this, I would like to thank Dr. Suji Akarwal. You would like to say anything, Dr. Suji?
Thank you very much. It's been an honor to be here, and really I enjoyed my discussion with you all, and thank you very much, and look forward to work together probably in future.
Dr. Suji, we look forward when it comes, as you are working on different kinds of sequencing technologies to identify different things. So here in this participant, some of the researchers also there, if they would like to collaborate with you in those areas, especially different sequencing techniques. So maybe they will contact you. I will provide your contact details. Is that okay?
Oh, sure. Okay.
If you happen to drop by Transit in Malaysia, do come visit our university. It's a beautiful campus.
I would.
You're welcome here. We have guest house here, and you can come
Thank
with
you.
your family, no problem.
Thank you, sir. Really an honor. Yeah, thank you.
Okay, so.
Thank you very much. Yeah.
It's too late for you to sleep. Thank you so much for your sacrifice.
No, don't worry. It's been an honor. Yeah, thank you. Thank you, Suresh, for this opportunity.
Thank you, Suchi. Have a nice day. I mean, we sleep well.
Nice day to you all. Thank you. Bye-bye.
All the participants, don't forget to fill the feedback so that we will provide you the e-certificate. Thank you. Bye-bye.